everybody.
Doug, are we good?
Yep, we're good to go.
Okay.
Welcome, everybody. It's an honor to be here today. I'm excited to kick off tonight's Think, Innovate Fireside Chat with someone I've known for many years and respect greatly, hosted by someone else I've known for many years and respect greatly. Innovation is really the heart of where we are here in DNA Valley in Montgomery County, Maryland. So this is going to be a really exciting conversation. My name is Chris Thru. I'm the founder of BioBuzz Media. We're your life science media partner for life science ecosystems. My team likes to say that we breathe the life in the life science communities, which is very cheesy, I know. But we do, someone else that breathes life into community is Jeff Galvin. When I first met him in 2018 at a similar event to this, right away, his energy was contagious, and since then, there's been no other person in Montgomery County that I've met that's been more of a champion for community and champion for innovation in this region than Jeff. So I'm excited to hear some of this conversation tonight. Innovation, again, is part of where we're at. It's an exciting time for this to be here. We're the birthplace of the Human Genome Project. Cell culture was developed right down the road at BioWhitaker, and now we're in the precipice of curing HIV here in Montgomery County, Maryland. So a lot of exciting things happen here in DNA Valley. So tonight I'm honored again to help MC this really exciting event and welcome you all tonight. Before we jump in, I want to pass it over to someone else who is no stranger to building thriving community here in Maryland. Kelly Schultz, CEO of the Tech Council of Maryland.
Thank you so much. Thank you, Chris, and gentlemen, Jeff, Mike, thank you so much for having us here today to share in this very special moment. American Gene Technologies has been a great partner for the Maryland Tech Council and for life science community overall in the state of Maryland, and it is so important for all of us to recognize those businesses that are working to cure the incurable. And I would like to give special thanks to Jeff and congratulate you on your upcoming recognition. But I also want to tell you a little bit about the universe in which we live in, not only here in Montgomery County, but also across the state of Maryland. Maryland employs, through their businesses in life sciences, 54,000 Marylanders. That's a lot. And the revenue that is generated from those businesses is upwards of $20 billion a year. That's a lot. That's something to be recognized for all of us, and when we talk about building the ecosystem, it's businesses, it's research, it's organizations like AGT and all of the others that are out there every day making a difference that's helping to create even a larger ecosystem and putting Maryland on the top of the list for places where businesses want to go and to procure and to continue to cure the diseases that are unknown to us today. So I want to thank you. Chris, I'm going to give it back to you. Maryland Tech Council, if anybody is interested, look us up on the web. Thank you.
Well, without further ado, let's get to what we're all here for tonight. I'd like to introduce Jeff Galvin, CEO of American Gene Technologies, guest of honor for this evening, and Mike Binko, who's founder of Startup Maryland and venture partner at Think Ventures.
Thank you, Chris. So I'm delighted to be here, Jeff, because I've been watching your activity, your enthusiasm, your energy in bringing some perspective to a well-established industry here and bringing some fresh approach and some use of innovation to this. Attracting and retaining a team and bringing a cure to a stable stage where others can get behind it. So I want to thank everybody else for joining us. We have some folks here, peers and stewards of the ecosystem of life science, bio, pharma, and related technologies here in Montgomery County in Maryland, also from across the river and actually across the pond from Belgium. Those of you joining us live online on the YouTube stream through LinkedIn, thank you also for joining. We have quite a few topics we want to hit on. We won't keep you too long, but we wanted to kind of highlight some perspective of Jeff's personal experiences, but also what he brings to the industry that he's chosen to lead and the organization that he's brought to market very quickly, swiftly, efficiently, and effectively. So Jeff, if you're ready, I'm going to hit you with some questions here.
Sure. Thanks for having me.
You bet. So American Gene Technologies, you joined the organization somewhat into its life cycle. Could you share a little bit of perspective of how that happened and how you became involved with AGT?
So I joined the biotech industry certainly when it was rolling, but actually American Gene Technologies started because I met Roscoe Brady at the National Institutes of Health, and he showed me viral vectors. So he was working on lentiviruses, and he had cured a disease called Gaucher's disease, which at the time, the cure that he fashioned was from enzyme replacement, which was similar to what Genentech was doing with making human insulin. Anyway, he made a missing enzyme for people that were dying of this disease, and it cured them. It didn't cure them really, but it treated them. It treated them in a way where it was no longer a death sentence to be born with Gaucher's disease. Anyway, he thought, rather than enzyme replacement, he thought, maybe there's a simpler way to do it. We're actually making this enzyme in human cells in a bioreactor. Couldn't we just reprogram the human cells in the bloodstream and let them make the enzyme that they're missing? And so he started working with lentiviruses, but he got to sort of late in his career, and there was some politics around some new rules at NIH that wouldn't allow people to both head a lab and be a consultant, and he was a consultant at Genzyme, and he had to pick one or the other. And so, long story short, they were going to close down his lab when I met him, and they weren't going to do anything with lentiviruses at the time, and he was the cutting edge. And for me, coming out of the computer industry, my head just exploded. I was like, what you just showed me is the diskette for the organic computer, the human cell. Now that we can reprogram DNA, is there anything we can't cure? And I said, I think you're about to throw away the most valuable thing I ever saw.
So the term you mentioned, reprogramming, share a little bit of your perspective coming from the technology and software space in Silicon Valley and what you associated with the opportunity in life science gene cell therapy.
Well, my perspective on humankind at this point is that each one of us is somewhere around 37 trillion collaborating organic computers called human cells that collaborate on a network that's electric, or chemical or physical, and that make us. And remarkably, every single computer on this network runs the same operating system, DNA. And the DNA is very similar between different humans that you meet, right? Almost exactly the same. Really what gives us such a variety of humankind is the expression levels of genes. But we have mostly the same ones. So what is the DNA, right? It's made up of these genes, and the genes are instructions to the cell to make very specific enzymes or proteins that then combine into other things and implement everything in our body. And quite frankly, it's the most sophisticated computer on earth. It makes, digital computers really look pretty barbaric or unsophisticated. In that, think about your human cell has an instruction in there, a gene that says make a copy of it. Wouldn't you like a command like that on your iPhone? It would save you a lot of money, right? And that comes from the fact that everything, even about how we develop from, just single cells to what we are, is coded in that DNA. So, why aren't we doing more with that organic computer? Well, the answer is it didn't come with a manual. So this is where the Human Genome Project comes in, right? Why did we spend $6 billion to go ahead and sequence 2 human genomes? I mean, we did learn something from it. We learned the number of genes, the number of base pairs in them. We actually successfully sequenced it from end to end. That was a remarkable thing to do, but it was the act of doing it. It was the moonshot of doing that that really yielded so much great technology that has been the fuel, for starting what I call DNA Valley right here in Maryland, right? Because what we have is a explosion of sequencing technologies. All the money that went into that brought down the cost of sequencing, the cost of PCR, the cost of reagents. It got everybody familiar with viral vectors that allowed us to do all sorts of experiments in cells that would give us more information about how cells work at the molecular level, how all the genes are related to, things in the cell and how cells actually interact in the body, because now we can do all sorts of experiments really, really easily and cheaply that used to be tremendously expensive and, and complex. So, that is the genesis of this industry in a way. That plus, think about how many PhDs were trained at both those labs in all of these technologies. And then what did they do? They just sort of, when those things imploded or exploded or whatever, all those folks were inspired to go on and create startup companies. Because back at the Human Genome Project, they used to say every time that they sequenced a new gene, there's a new company.
A virtuous cycle of entrepreneurship when innovation is applied and organizations can grow and folks get the confidence to then go do it themselves with things they've learned or brought or new directions they want to take it.
Yeah.
So with that Silicon Valley heritage, taking AGT to market as a gene and cell therapy leader, the code compile, reprogramming, the human body, the human genome. I mean, a lot of that was done here over the decades that the effort was put behind it. A lot of science-driven research, a lot of federal labs for the U.S. government nearby, a lot of willing entrepreneurs, and some resources on the investment side. Could you share a little bit of your perspective on kind of navigating AGT through a little bit of the waypoints that it took to kind of innovation roadmap the first cure assay that you went after? And could you share a little bit of where that's manifest recently with HIV cure?
So the arc of the story is I meet Roscoe Brady and I see this thing and I have a vision, like an epiphany for the future of pharmaceuticals. And it's so exciting that it reminds me of when I first saw computers in the 7th grade back in like 1970 something. And after learning to program, I realized, wow, these things are going to impact every life on Earth. This is just going to relieve people of so many rote activities with words and numbers and stuff like that. It was just crystal clear even to a 7th grader at that moment. And then I lived near MIT, so I used to wander around the campus and I saw robotics and sensor technology, and I could have told you back in 1970 that eventually computers will do everything for us, including driving our cars. And you would have called me crazy back then, but sometimes you see something and you realize what it will become in the future. And that's what happened with viral vectors. Is I said, okay, if we can modify DNA, we're really down at the root drivers of everything in the human body. And having had the experience in Silicon Valley, and having been part of the computer revolution, the software revolution, the internet revolution, I mean, these are just all technology curves that I had the fortune of being able to be involved in. And as a matter of fact, getting to the point where I had done so well in all those technologies that I was retired. So I just said, this is too exciting to not get involved in it. And of course, being an entrepreneur, I had to start a company around it, and NIH was willing to just let me have everything that was in Roscoe Brady's lab, including the postdocs. And so I started the company.
So being an entrepreneur, you don't just get involved sometimes, you actually have to take it all forward.
Well, like you may imagine that you could find an easier way to do it, and maybe there is an easier way to do it. But what I would say is that, usually the visionary leader has the drive to go ahead and keep pushing forward under all conditions, and that of course will help to bring the entire group along. That depending on how clearly people see that vision, or at least feel it when it's articulated or whatever, will give you some sense of their ability to commit to it and stick to it and contribute to it. Of course they have to have skills and all that stuff. So I think for me, it turned out that, I had to drive this thing, because at first, I just hired a bunch of people and I said, go forth, implement my vision. And, that worked up for a couple of years. And then I just realized this thing just is falling apart, and I moved here pretty much full time. And it was just put a ton of money into it at first. I was believing in it. I was running through my entire retirement.
Personally, as
Personally.
the primary investor in the company.
Yeah. And, you know, finally, you know, I felt like this thing would go through its dot-com boom.
Mm-hmm.
Eventually what would happen is that, you know, people would understand the power of gene and cell therapy, and they would see so many things going on that, you know, the usual suspects would start just minting companies, right?
Economics might take over and amplify.
Yeah, I thought, you know, what will happen is Wall Street will jump in, they'll overvalue the companies, you know, and then there'll be 1,000 gene and cell therapy companies, and, you know, 1 out of 100 of them will actually have a product that could justify their value. But then I thought about it a little bit more, and I said, you know, even if you were to create 100 companies in this, most of them will backfill their valuation, provided that the disease they're working on and the mechanism of action, you know, sort of the gene that they think is operable in there, they'll probably eventually get there if they have enough money. This is something to remember about the gene and cell therapy industry, is that, you know, it's like software, right? We're making little genetic constructs that improve health by going into the cells and, you know, rewiring that operating system from the ground up. It's sort of the difference between you know, putting new software on your computer, and, you know, an old drug would be like pouring water on top of the computer and, you know, seeing whether you get the result you want. Well, that didn't work, let's try orange juice, you know, let's try, because, you know, that's the way normal drugs are, they go all over the body. But now we can take these viruses, crack them open, you know, we can scoop out the diseases that are in that virus and throw it away. So those are little DNA constructs. Then at this point, the industry's gone so far that we can order any human gene over the internet. It'll come the next day by FedEx and a plasmid. We can chop it out, we can put it in some of these and hollowed out viral shells, and we can infect some cells and see if we can turn on that gene, and we can start to see what the effect would be as well. So, you know, this is, you know, what we're now able to do is to, you know, make very specific changes to that, you know, the instructions in the cell. And the viruses themselves can be tropic to certain cells, so we can narrow it in on just certain tissues, and that reduces the number of side effects. So now you're seeing that more drugs will actually make it through the, you know, the regulatory process, because, you know, what kicks a drug out of the regulatory process? Well, of course, it does something good, but then you find something bad that it does. Well, with a molecule, how do you get rid of the bad thing? Well, with software, how do you get rid of the bad thing? You debug it.
Yeah.
Okay, and there's been examples already in gene and cell therapy, like GSK had a cure for ACA SCID, but it was causing leukemias, and, you know, the EMA wouldn't approve it in Europe, with leukemias being one of the side effects, right? But it was a very important cure because it was curing bubble boy disease. You know, people with no immune system could, you know, get this treatment and live a normal life. Well, it turned out there was just one little problem in the 3' LTR, you know, a little bit of code that was causing the leukemia. They chopped it out and they debugged it. It's sort of like the difference between, you know, if Microsoft, you know, when they introduced Excel, it probably had a few bugs in it, right? Well, if that was traditional drug, you would throw it away and start again. But if it's software, you can find out what the bug is and you can change it. So all these companies can actually have all the code.
Yeah.
And also what you learn on that software will help you on the next software, right? So, you know, this is the industry that we're in now. So I imagine that it would be explode like that. And it took longer than I thought. But sure enough, around 2013, there were other people that were willing to invest in my idea. So it wasn't just me carrying all the burden myself, but, you know, I had to go out there and start raising money.
Sure.
Yeah.
So a little bit of what you talked about is, like, you saw a confluence of innovation or technology or approach that could be brought to an industry that was very established, very somewhat rigid, regulated. You know, in the internet world, we used to call telcos dinosaurs. So some of your perspective on kind of changing the industry for its own good is, you know, some people have thoughts of, do you try to change the established environment, or do you take a Buckminster Fuller approach, and if you want to improve it, you actually create something that makes the existing model obsolete? It seems like a little bit of your perspective is that, you know, we don't need to necessarily pay attention to the established way of doing things. We can apply innovation and take it in new directions.
Well, so if I was going to give advice to CEOs that were going to go into gene and cell therapy, my first advice would be forget everything you know about biotech and pharma. And coming out of the computer industry, the good news is I was probably the perfect blank slate to go into gene and cell therapy because I wasn't held back by a lot of common wisdom about the right way to do things. So that's, why I'm doing things differently. I'd like to say it's like, well, I did all the calculus and, I realized like, here's the formula that will, do this. But no, what it was I came from a different industry. And of course that outside perspective did lead to a lot of ability to sort of predict the technology curve that I would experience again in a new industry, even though it was, only related by the fact that the way it operates is similar. Software, for the human computer, software for the digital computer. There's a lot of parallels in there. So right guy, right time, right place. Just enough money to survive until I could get money from somewhere else. The passion to keep going on this thing, and so it wasn't such a calculated approach. But I would say that what I did contribute, and maybe I still contribute to this industry somewhat, because now for 15 years I've been telling everybody my vision, so hopefully some people have picked it up. But I found ways to leverage things that I've learned in other technologies to create efficiencies in this industry that I think will have huge benefits to patients and to companies down the road. Because, in the old drug development industry, there weren't really reusable components. Now in this industry, you can actually invent things that could be part of many different drugs, and they can become building blocks that can get you to new drugs in the future. And what we've done at AGT is create a broad platform of those things. And I like to say that, if you do a thought experiment at AGT and you realize that by reading what's on the NIH website that, here's the molecular biology of this disease, and you go, if we just upregulated this gene in these cells or downregulated this gene in those cells, we could probably mitigate the disease. And I'm talking cure, because, a lot of diseases are just, one thing's missing. You put it back in, you're just not diseased anymore. That makes a cure, right? All right, so you do the thought experiment. Well, here's all our components. Is it within the scope of our current component technology to deliver that gene to the correct cells in a targeted manner so it shields the healthy cells from effect safely, right? All right, you can start on our platform and you're 80% or 90% done with your drug day one. All right, so you save 90% of the cost, 90% of the time, 99% of the risk. Well, one day, I think we can convert this into a cottage industry where hopefully anybody can cure a disease that they really have passion for and knowledge of. I think even parents that are born, who have kids that are born with monogenic diseases that are unaddressable now, in the future, it will be within their means, without having to raise millions and millions of dollars, to actually develop a first human efficacy of a drug that mitigates their own kid's disease. Well, certainly right now, there's going to be tons of college professors out there, tons of scientists, tons of doctors that, have a lot of knowledge about a particular disease and are really excited about it. And if they come, we're not prepared to do this right now, but eventually we're going to open our doors to them with all our components and go, come tell us about your idea. And we're going to sit down with them and go, yeah, that could work. Then we're going to give them the platform for free and just go, pay us on the back end.
Yep.
If you ever make any money off of this, we just want a piece of it.
I think that's a very interesting perspective that you've shared vocally with the industry, that it may be coming. To get there, you had to secure methodologies, art, intellectual property. And also in our pre-interview, the patent wall, as we kind of effectively refer to it, is substantial and it's impressive. And over the years that you've been building AGT, that is going to be a framework that these other startups and companies can I love these metrics that you highlighted: 90% cost reduction, 90% time saved, 99% of risk. For a heavily enforced regulated sector, that risk factor is often the limiting factor of entrepreneurs actually having the confidence to go, particularly if they are very credentialed, long decades career peer review, and there's risks in kind of doing some things.
Interesting.
Do you think that perspective of you as a kind of technology, maybe software, various technology entrepreneur, where you may have not come out of that peer-reviewed, research-driven experience, gave you maybe, I don't want to say the naivete, but the ability to kind of look beyond that and say, this has to happen for this industry to evolve?
Well, look, saying that I was naive when I got into this is not a stretch at all, and not insulting at all either. And if you talk to my wife, she'd be like, yeah, we came this close to being, destitute, thanks to, his naivete and his willingness to jump in when the risks were extraordinary. Now the risk is, it's diminished now that we've got so much momentum. So in terms of whether it affected me, yeah, well, of course, I benefited from the fact that I didn't know what I was getting into. Yeah, that's clear. And that my vision was correct, even though I misunderstood how long it takes to do something in biotech. So any biotech person would have been frightened away, any pharma person would have been frightened away. And I think there is something true about the idea that, when you're in an industry where your peer-reviewed journal articles and citations and awards and stuff like that is your whole credibility, yeah, you might want to be more careful. But I also think that the reality of it is that even though they may have exaggerated the idea of how much it costs to create a new drug, maybe it's not really $1 billion or it's not $2.1 billion like some of the studies show, it is a ton of money, because when you're in the type of drug industry where you have to discover and test, and anything can kill it, right? And you have to tip up like a pill factory for the phase 1. So you can treat 15 or 20 patients, and you can get a side effect in that. It's like, okay, bulldoze the pill factory. It's not good for anything else anymore. We just spent $50 million on that. That's the old drug development. Now it's all done in a cell culture hood, right? And the translation between mice and humans is amazing. If we're talking about a homologous gene between a mouse and a human, it's fairly predictable. And so it's a whole different sort of risk factor there and cost factor. And then this was part of my vision as well. I thought the FDA will get smarter and smarter about this. Like, why the heck would you make everybody test every single thing in every single way when you have data from all these different experiments that may have already proven one thing or another thing that you can apply to the new drug, and you could actually one day, I believe the FDA will simulate the body in silico. In other words, a computer simulation will basically simulate us all the way down to our molecular biology. And then they'll receive the drug over the internet in the, coded in the ACTGs. So you'll just send the sequence of your drug there, and they'll go ahead and test it against, 1 billion genomes. And they'll go and they'll say, well, it looks safe except for we don't know about this, this, and this. So if you can give us a good theoretical explanation for why it's not going to do this, this, or this, you can test it. So what I saw was that whole regulatory barrier was also going to come down. And you can already see in gene and cell therapy, it doesn't make sense to do thousands of patients, to decide whether to, release a drug that is for a deadly disease that only affects 1,000 patients. Right? This is the reason that those drugs don't even get done by pharma companies, is because they can't spend $2 billion and maybe lose it all, on something where they're never going to sell more than, 10,000 doses in the lifetime of that drug. But if you could develop that drug for $10 million, tell me a disease that isn't worth curing, right? We're going to be talking about 7,000 monogenic diseases falling to gene and cell therapy. This is just inevitable. And it's going to be a matter of all the forces that are at work already, coming together and creating an environment where all this stuff just naturally occurs.
So does cost, time, and risk, to me, if you're getting to the 99% elements of those, confidence from resource providers increases. Direct correlation.
It should.
And I think what you've done with AGT proves that out. Full disclosure, I did get involved as an early investor in a round right before the clinical trial. And I think the folks that I brought in were through a syndicate. And I think it was the D round. And everybody that I had in my syndicate were data scientists, data analytics, cloud folks, and software folks. And when I shared with them the prospectus and the opportunity, they immediately leaned in and said they saw some of your interviews and code, compile, review, recode. Struck them that this industry is rife and ready for this type of disruption in a positive way. Now, you look at any new technology or innovation that's out there, and you've talked about several that you've leveraged through your experience of implementing them in other sectors, and they now come together and can amplify. Do you see investor confidence in this space finally making a turn? AGT, I think, is proving things with, your success in kind of finally getting out from under having to fund it yourself. Could you share a little perspective of that? Because I'm sure it was still fairly tight, and attracting, retaining talent is not cheap in this area, competitive market.
So, boy, there's a lot to unpack
know,
there.
sorry.
Okay, so it's okay. So, the attracting and retaining talent, we've been very lucky because we have a very exciting story, and that's because it's vision and mission driven. Working at AGT reminds me of the early days at Apple. And actually the way that I attract everything, including the money and the employees and the collaborators and everything, is by being what? An evangelist, right? We used to use that term at Apple because we were evangelizing something that was a better way to do something in an existing industry. And you go out there and you tell anybody that's willing to listen. And then the ones that sort of get it, they feel it, right? Because they feel your passion, they feel your belief, they get excited, and it's not just an intellectual connection at that point, it is what creates movements, right? And so it's those people that feel it that come and help power the thing with you. And like what you were saying is that, my metaphor that I was using in a lot of these videos about, the organic computer and, the operating system of that computer and stuff like that. Yeah, for people that had already been successful in software and computers, they could, intellectually attach to that. But when you see what can be done with it, when somebody tells you like, this is where I think it's going, and tell you that story, well, then you're emotionally involved. You might want to invest not just because you might make money, but you might want to invest because you want to see that future and you want to see it as fast as possible. Cures for everything, even all of the terminal cancers are eventually, going to become within scope of gene and cell therapy, where we're going to send radiation and chemotherapy the way of bloodletting and leeches. And who wouldn't want to be part of something like that? And, even VCs, right? Because you were saying, well, did people catch on? Well, the answer is yes and no, right?
Some.
So individual high net worth individuals, and angel investors, angel funds, small VCs, okay, they could, catch it. But the institutional investors, not so much, because they run a model. Okay, so, how do you get rich in traditional investing? You find something that works and you do it over and over and over again, and you don't break that model, because when you go outside of that model, that's when you lose money for the first time, right? So you'd rather make a little money reliably on every deal than you would want to make a lot of money on something that you just have this gut feel like this is going to be huge, right? So institutional investors take a little bit longer to convince. I'd say they're starting to get convinced, but I would say for people that are starting up in gene and cell therapy, it's still difficult if you're not like the traditional VC-backed company. So what would that mean? It's technology that's spun out of a brand name university from a brand name lab with a brand name, scientist that has done something before that's of note, and a CEO that's made money for that VC a few times over. And they'll pick somebody from Pharma and Biotech over somebody from software in a split second, right? Because that's who they're going to believe in. Okay, so then they've now minted a company that, I don't know whether it's got the same level of creativity as an AGT, right? But at the same time, all right, it fits within their model and they feel like it's likely to succeed, and they've got, an essential element of that, which is lots and lots of money. Okay, so we came at it a different way. We were raise and spend, but we kept on making progress and then showing that progress and raising some more money and then making more progress. And this is how all the milestones happen at AGT. So we grew organically that way.
We see that in some of the new perspectives on early stage and growth capital of it's no longer industry investing. It's innovations that can prove themselves in maybe one or two industries. And there's a diversity, there's a risk factor there. But if they can really start to kind of gain the traction where the industry is starting to lean in on the innovations, So we talked a little bit up front because I was curious of your perspectives on things like artificial intelligence, certainly machine learning and others. It sounds like that metric and milestone approach that you've taken with AGT and amplified in the last couple of years with outside resources increasing, the team amplifying, is that there's a few select innovations that are getting the dialogue right now, but you might not be confident, or you might not be as large of a proponent of some of them, like AI. I've kind of heard you talk about, not yet, but I know there's a confidence that you could probably make that stick. The economics are gonna have to pan out on that.
Well,
Curious.
so I'm not quite even sure what you're asking me there.
Do
but
you feel AI
inspire
is-
some thoughts? Oh, so you want to
Is an
know about AI?
impact for what AGT has done or can?
You know, I don't want to. I think AI is overhyped right now. I think a lot of the attention being paid to AI is because people don't want you to pay attention to the dangers of social media.
Mhm.
So AI is the new boogeyman, and we don't need to worry about the fact that your kids are being destroyed by Facebook, you know, and Instagram and TikTok, right? Okay, what we need is to think about managing the problems which are real time and current, and AI certainly is going to have a whole lot of challenges to it. But should we change all of our thinking and only look over there? No, I don't think so. And now, what can AI do in our industry right now? Of course, you know how I can increase the value of this company is to go, we're now combining AI with it. Why? Because when you don't understand technologies, what do? You look for proxies to decide what's going to be successful. And that's how, you know, much of the industry, as you say, including the investment industry, looks at things, right? And so, you know, if something's hot, it gets a lot of money. And if it's not, it doesn't get money. But, you know, you don't sort of, Rome isn't built in a day, right? You know, it's built, you know, sort of one piece at a time, and you start, you know, it builds up. And that's really how companies should build. But, you know, you think about, like, when you get a tremendous amount of money, it changes the whole picture, right? It creates a new way of succeeding in the money world, which may not even efficiently promote value in the real world. Anyway, so AI is super hot.
Yep.
You know, invest in it, don't invest in it, do whatever you want. It's useful right now because if we're writing something, we want the AI bot to take a look at it and go, oh, you know, dumb this down for us, or, you know, make it seem snarky or more confident, or, you know, because it's just language models. That's all AI is, right? And it makes you think that it's really something special because it can imitate so well, right? Is it really thinking? No. Right? It's artificial for sure. Is it intelligent? No. Now, could it eventually become intelligent? You know, we can talk about that for hours and hours and hours. But right now, I'd say it's definitely high on the artificial, and it could be used in a lot of, you know, positive and negative ways. You know, it's like every technology; it's a two-edged sword. Okay, so AI, not so much. But remember that every revolution powers the next revolution. So maybe AI isn't ready to power, you know, the gene and cell therapy revolution just yet, but guess what is? Computers, software, robotics. So if you look at gene and cell therapy, why is it getting, you know, so great, so fast, and getting cheaper and cheaper so quickly? Well, that's because it's like Einstein's, you know, train on top of a train on top of a train on top of a train, right? So if you were to look at gene and cell therapy and go, Moore's law would apply here, I'd go, you're absolutely right, except for it's way faster than Moore's law because it's riding Moore's law. Moore's law is in the layers underneath, and then now gene and cell therapy is on top of that. It's at near light speed, right? So, you know, a lot of people are like, well, you know, gene and cell therapy, like we're working on a cure for HIV. And sometimes I get approached at presentations, they'll be like, but what about Africa? That's where all the HIV is, and you're proposing, you know, a cell therapy, and you know how expensive those are, and, you know, okay, I get it. But you see, I'm confident that if I can cure HIV in the United States, it's going to go to Africa eventually, the same way that I knew that computers would make it all over the world, and for the same reason that people in sub-Saharan Africa are carrying computers in their pocket that are more powerful than the biggest mainframes in 1978, right? You know, that is the nature of technology. that curve, that Moore's law will apply to this, and then the question is, well, how fast will it get there? And the answer is, well, probably faster than the computers did. So maybe we're 20 years out from a shot in the arm that cures somebody of HIV in sub-Saharan Africa. I can't tell you for sure, but when it'll happen, but I'm pretty darn confident it will happen eventually. Predictions don't even matter at this point. What matters is every journey that we're going to take is going to start with a single step, right? We're just trying to make that first step here, prove out, the idea of gene and cell therapy, that it can have this major impact. Then we're going to prove out that the idea of the platform could make it, more prolific, that more people could get into this and make more solutions. And then it'll just happen on its own. And then we'll see when it happens.
So you've talked about several disease tracks. Why HIV first? I mean, you've just touched a little bit of it, that the confidence was there and some of the heritage of the research that had been done, but why HIV first?
So people are always asking me, like, why did you choose HIV as your first product? And I say to them, I didn't choose HIV. HIV chose us. Because what happened is that, when you're developing a platform, so let's say you've got Microsoft DOS, right? You don't decide what to write. You look at what you got and you say, what might work on top of this? And, if you're not sure what would work on top of it, what would you do? You'd try a bunch of stuff. And, if you found something that worked, you'd bring it out.
Spreadsheet or word processor, right?
Whatever, right? Microsoft Flight Simulator,
Yeah.
who knows, right? So that's what we did with this. I said, okay, I got a platform and it's getting better and better, and so the scope of it's getting wider and wider, but let's just do experiments and see, what it works in. And we tried prostate cancer, and we got some initial results, and we tried liver cancer and hepatitis C, believe it or not, and HIV and phenylketonuria and familial dysautonomia. And if you've heard of that last one, there's only 400 people on earth with that last one. So, I mean, we were willing to experiment in anything because it was relatively inexpensive to do the experiment, because it was just in the cell culture and in cultured cells. And that was pretty cheap to do, and it needed a PCR machine and and some viral vector, and we could do all those things in a small lab. So, we get out and we're doing all these experiments, and you start seeing some results where you're like, that's actually intriguing. So, in phenylketonuria, we had a couple of patents and some technology that looked really promising. In immuno-oncology, we also had some of that. But in HIV, we got a 4-log reduction in viremia in an HIV challenge test with our first idea out of the chute, which, by the way, I thought of in my bathtub back in 2010. I was sitting there going, what do we do for a living at AGT? What we do is we crack open HIV, we scoop out AIDS and throw it away, and we use that delivery vehicle to deliver things to cells that can upregulate or downregulate any gene. And then I'm like, well, wait a minute. The only reason HIV is so hard to cure is because it's genes that are integrated in your cells, so turning them off is tough, but that's all we do, and we're doing it with HIV, so what are the probability that we could get into the same cells? Pretty high. So I thought, okay, let's just put knockouts to HIV genes into an HIV shell, lentivirus, and let's treat patients. Now, I'm not a molecular biologist, I'm not a geneticist, I'm not a cellular biologist, somebody that understands all these things, but my PhDs were. So I went to them and I said, what do you think? I mean, would that work? And they were like, yeah, we could pick conserved regions of the HIV genome and do that. That should work. And I'm like, all right, I helped. It's kind of exciting. And then, of course, now I'm really into it, and I'm reading about HIV, and I read about the Berlin patient, and I realize, okay, that person just has CCR5 missing, and I'm like, well, we can knock out the CCR5 gene, but if you've got matured T cells that already have CCR5 on them, would that fall off? So I go into the PhDs again, they're a great resource, and I say, if we knock out the CCR5 gene, would these T cells that are already fully matured, would they lose their CCR5? And the guy says to me, sure, the outside of the cell are just lipid rafts. If you don't refresh the receptors constantly, they'll disappear. So if you knock out the CCR5 message, which is how we knock down genes, you should end up with cells that are CCR5 negative. I'm like, put that in the project too, right? Because this is another protection that allows the immune system to actually work against HIV.
So an iterative approach that you're not trying to boil the ocean with this approach to assaying the cure. You saw some opportunities to apply what you had as
Yeah,
your own.
exactly. Well, no, it's just creativity, that's it. And that's why we say at AGT, creativity cures. It's just thinking out of the box, right? It's not saying, well, HIV is this disease that's a blood disease and we gotta kill every copy of HIV in your body or kill every cell that's got a copy of it. No, it's saying, what do we know? And is there a little creative thing we could do on top of the Berlin patient? Because the Berlin patient is cured of HIV, or was, he passed away of something else. But the point being that he received a bone marrow transplant that all it did was remove all of the CCR5 in his immune system. And then because he had an R5 strain of HIV, in other words, it needed that door handle on the cells to get into the cells, couldn't get into the cells, so the cells couldn't be infected and depleted, so they could do their job. What's one of the things that they do? Your HIV T cells clear HIV. It's actually easier to clear than a cold, right? It's a slower moving, more fragile virus. And so, the only reason that it gets into the body is it has evolved that one advantage where when it meets that sentinel T cell that's supposed to kill that virus and instruct the rest of the immune system to then go out and find the rest of it, it infects that cell, the CD4 helper T cell, the general in the immune army, and converts it into a soldier in HIV's army, a viral factory. And it never calls out the protection. Never calls the Air Force, it never calls the foot soldiers, and HIV depletes all those generals, and then it can spend 5 to 10 years depleting other generals, and that's what we call AIDS, right? Okay, so, the CCR5, removing it in that person's body, since he only had CCR5 version of HIV, it couldn't move anymore. He didn't need antiretrovirals every day to suppress the virus. He was functionally cured. There may have been some HIV in his body, but it didn't matter. It couldn't infect anybody, and he couldn't get AIDS. That's what he cared about, right? And actually, he couldn't recontract the R5 version of HIV either. All right. So I said, okay, we can do that without a bone marrow transplant, because 6 out of 10 people that go to get a bone marrow transplant die from the bone marrow transplant. If you've got HIV and your choices are take a pill every day for the rest of your life, or take a 4 out of 10 chance of surviving, right? You're probably going to take the pill. Okay, so we could bring this bone marrow transplant effect to people without a bone marrow transplant. That was the whole idea of the cell therapy. But we could make it, the next creative element was actually the first creative element, which is that we could make it work against all strains of HIV, because we could find a short interrupting RNA. Basically, it's a little teeny, sort of sequence of nucleotides that will bind to some part of a genome and prevent it from doing anything. So those siRNAs are protective against all known strains of HIV. So actually, we could not only give, the equivalent of a bone marrow transplant to somebody, we could do it better, cheaper, faster, and, and way safer, right? So that's a product, right? 38 million people on Earth are dreaming of never thinking about HIV again.
And that's a great why.
Yeah.
That's a great why, HIV.
Yeah.
So the last 18 plus months with your HIV clinical trial, pretty monumentous. Could you share a little bit of perspective on kind of the results that your team achieved?
Gladly, yeah, I'm very excited about this. So we have a successful phase 1 in that we got 0 serious adverse events. Of course, that's the primary endpoint of any phase 1. And so 100% on that, you can't do much better. And then there were secondary endpoints, because gene and cell therapy is such a profound effect on someone's body that may never wear off. The FDA doesn't allow you to even test these drugs on anybody that doesn't have the disease. It would be unethical if there wasn't some potential benefit to those people to go ahead and change them forever, right? You're not going to take healthy college patients and reprogram them and see if there are side effects. You're going to take people with the disease, and only if the FDA decides the disease is important enough that it's worth the risk, right? All right, so we have HIV infected individuals, and we put this drug in their body, so guess what? We get some level of efficacy readout as well. So in the phase 1, we got what's called blood markers of efficacy. These cells that we had modified to actually fight HIV, we could see that they were doing it in the body. They were essentially engrafting. That just means that when we put these cells into their body, they found a home. They were persisting. In other words, the body didn't look at them as foreigners and eliminate all of them. And they didn't, have any adverse events where they attacked the body, right? That was the safety result. And after even a year of these cells being in the body, unstimulated, because people stayed on their antiretrovirals, we could still find those cells. And they were still reactive to HIV. They still maintained their characteristics. Okay, so that was important, right? That's blood markers, sort of a theoretical, sort of representation that it's working properly in the body. But what's the real test? The real test is you take away their antiretrovirals and see what sort of efficacy you get when it comes to viral suppression.
And when that happened, when did that happen in this clinical trial?
So right after the phase 1 was done, we did an IRB approved study. So we sent this to the FDA. They didn't comment on it, but the IRBs approved it. And so that meant that the FDA wasn't going to stop it, and the IRBs thought it was safe. And so we asked these patients, hey, you've got some of these cells left in your body. We'd love to see how well it suppresses the virus. Would you be willing to go off your antiretrovirals? And so 6 out of 7 of the patients actually agreed to go off their antiretrovirals, and 100% of them showed an effective immune response. And what I mean by that is, instead of, when you take them off of antiretrovirals, what should normally happen if they don't have protection in their body any longer is the HIV virus comes out of the viral reservoir, and then it will deplete the CD4 cells, these helper T cells, the generals of the armies, and you can just measure that. That's why, when they diagnose somebody with HIV, you always hear, what's your CD4 count, right? So the CD4 should go down, and the CD8s, because the generals are dying off, they never call out the Air Force, that's the CD8s, you shouldn't see the CD8s go up. What we saw was the exact opposite of that. The CD4 stayed stable, as if they were resisting the viral attack, and the CD8s went up, as if the generals were surviving and calling them out. 100%. So that was our first really exciting result, but it got even better. Because we said, well, the first time you see the virus, these cells have been quiescent. They've been like resting in the body for a year. They're really not ready for it. Kind of like the first time you get COVID, right? Your body's not ready for it, and so you get symptoms. And, well, how do you avoid that? Well, you get a vaccine. Now what does the vaccine do? It looks enough like the virus that your body amps up the cells that would be useful in fighting that virus, and essentially it's called activating them or putting them in effector state. But you can imagine the generals are like proliferated a little bit, and the Air Force is on the runway, fully armed, okay, and fully fueled and ready to go. All right, now the pathogen comes in a second time, right? And you don't even get symptoms. Right now, what else vaccinates you against COVID? Getting COVID. The next time, right, you have about 6 months of protection, assuming you survive without any damage from the initial COVID, right? All right, because what looks more like the virus than the virus itself, right? All right, well, guess what? HIV's in the body of each one of these patients. We just expose their immune system to the HIV. And we can just put them back on antiretrovirals so it does no damage to their body, and they don't even get any symptoms. They're certainly going to take like years to develop AIDS. So we bring them back down to what's called well-controlled or undetectable, and we go, how many of you would be willing to come off a second time? Because if our cells are working the way we expect them to, we should get a really good result the second time we take you off of antiretrovirals. 100% again. 4 out of 6 agreed to go off a second time. And all of them got at least a 2 log reduction, so at least 100x reduction in the viremia. And half of them, so 2 out of 4, were at levels that they are considered to be long-term non-progressors if they stayed stable at that level. And they stayed stable for quite a while before we put them back on antiretrovirals. Why do we put them back on antiretrovirals if they were stable and long-term non-progressors? What does that mean? Long-term non-progressor means you wouldn't need to take antiretrovirals to prevent AIDS. So the only reason you'd be taking it is to prevent transmission, okay? So that's why they went back on their antiretrovirals, and we recommended it because it's important to prevent transmission, right? Safety.
Yeah.
But That is amazing, because normally a long-term non-progressor might be less than 1 in 100 patients, and we got 2 out of 4. Okay, so statistically that's exciting. And the other 2 were almost at that level. Close enough that you just believe it's probably just a matter of dose. Now, what do we need to achieve? We need to achieve an immune system that suppresses this virus at levels where they're non-contagious. So they're at about 5,000 particles per ml. Right now, that would be long-term non-progressor. And they did this on a 1% dose of their original dose. And what do I mean by that? When we infused them with the cells, they got about 1 billion cells. A year later, because these cells are not stimulated, they haven't seen HIV yet, so they have had no reason to create cytokines and to react and to alert the body that they're necessary or to proliferate or any of those things, right? So the quantity of them probably goes down to about 2% or 1% even of the original number of cells. And yet when we took them off of antiretrovirals, look at what we got. Well, if we took them off their antiretrovirals at the beginning of that year instead of the end of that year, like right after they're infused, you're talking about at least 50 times the dose. Is that dose safe? Sure, they got it already. Nothing bad happened to them. We got to think about, okay, what are the other things that might happen? And we need to explain to the FDA, okay, if we saw this, here's what we would do. If we saw this, here's what we do. Because there's no such thing as 100% safe. Any change to the protocol needs to be explained. But we think we've got, good rationale for why this test is reasonable. Well, if we pull them off immediately and they've got 50 times the dose, could they get suppressed down to undetectable in their first their first set of withdrawals? We'd still withdraw them twice. We're still going to expose them to the virus briefly, just to make sure that all those cells are activated in effector state, that they're ready to go. Then we're going to bring them back down to well controlled. Then we're going to take them off a second time. So we should get a result that is in line with what we got after a year, but hopefully much more potent, and maybe that that 5,000 could easily, remember, they came down from 500,000 to 5,000, right? That is, incredible. That's like 1,000-fold, but all of them at least, came down 100 or several hundred-fold. All right, well, they're pretty darn close to, between 5,000 and 50, which would be non-contagious. That's another 2 log.
So with this success with ATT 103-T, you're not stopping the business process of confidence that you're creating in your innovation. So could you share a little bit, you announced something recently with the HIV cure that is indicative of some things we talked about of kind of making AGT a platform that you can not only step and repeat a cure for a disease, but how are you going to approach multiple cures and multiple diseases? So could you share a little bit of the confidence that the business gained and maybe what the market showed confidence in AGT in looking at the HIV cure assay as an opportunity of its own?
So since HIV is going so far ahead of everything else in our portfolio, we think that it deserves a singular focus from a team and from financing and everything to try to push that thing over the finish line, because there's more work to do. It looks like we have good visibility on the goal line, and so I think the rational thing to do is to put a tremendous amount of rocket fuel behind that, and the way to do it is to spin it out as a separate company and find separate financing for it. And we're already partnering up with a very well regarded Wall Street firm and we're looking at a variety of different ways to bring in a really big chunk of money to make sure we have lots of gas in the tank, because this phase 2 is definitely worth doing. It seems highly likely to yield an even better result and maybe even across the finish line. And so, this is now investable on Wall Street. So, as I talk to you today, this is, sort of our vision going forward. We have announced that we're doing the spin out, and we are going to be starting a round of funding, just for Adammune in the not too distant future. And I think that what we'll see is an opportunity to prove 2 things, right? To prove that HIV is curable, But the second thing it proves is that we can efficiently create things off of the platform, and then, we can use some of that success in terms of, sort of attention on AGT, because I think that was going to interest a lot more of the, sort of traditional industry folks once we prove that platform idea with, I think HIV would be a blockbuster drug, cure for HIV. So once you prove it, but also, that comes with financial rewards as well. So I'll have more money that I can put into AGT, and I hope our second act will be to go on and cure cancer. I think that's going to take longer.
Sure.
There's still a lot of work to do. But in the meantime, we can implement this idea of letting other people use the platform, and maybe enabling a whole bunch of drug development from people that are smart and passionate about diseases and knowledgeable in that area that could, on the kind of money that you could raise in an SBIR grant, you might be able to cure or at least get an IND for a rare disease. And if you've got an approved IND, I think most entrepreneurs could probably find $10 million if that's all it took to get the first human efficacy. And remember, even a traditional pharma executive will tell you that if you have first human efficacy, you have an asset. If you have an asset, you can sell it to them.
Yep.
All right, so I
how
think there's a
many-
lot of hope there.
So how many other bathtub epiphanies could benefit from your yeoman's task of taking yours forward, and then they may not have the cost, time, or risk of taking it to their own market?
Exactly. Yeah. No, hopefully this will become a cottage industry.
Yeah.
The garage startup will be reborn in biotech. And I think that's possible. It's not going to be an overnight thing, but I think you're going to find that a lot of small companies are going to just rise organically out of, fertile soil like we have here in DNA Valley. From Montgomery County up to Frederick over to Baltimore, we have this area where you've just got the talent, you've got the the STEM education and workforce, you've got a huge amount of just entrepreneurial talent, and you've got an industry which is just in this incredible growth phase and revolutionary phase where we here right around the epicenter of it are feeling the excitement, and I think you're just going to see people go, I think I can cure that now. I think I can cure that now. And, they're going to go ahead and start on the journey. And, I envision a day where 2 out of 3 drugs that are thought of that way become actual drugs one day. That will be the reliability of, sort of the imagine, design, test, debug, finally get it through. Are going to yield, those types of ratios. You know what the ratio is now in traditional drug discovery? It's like 1 in 50,000 molecules makes it, to a drug. Don't quote me on that. Look it up on the internet. I'm not sure what the exact number is. But this is my speculation, is that, I think we're already at the point where, if you look out there in the industry, if companies have enough money to keep going on the debugging process, most of the companies are actually getting there. What's happening is a lot of companies are running out of money because it's expensive to test in the clinic, and so they need to learn better about how to anticipate the clinic in the cell culture hood, which is a way cheaper place to fail, right? But in any case, what's going to happen is everybody is going to get with it. Even the FDA is going to make it easier and easier and easier to go through this process. And I think we're going to just see, a drip, drip, drip of miracle cures. Like there's a cure for a type of blindness. There's a cure for spinal muscular atrophy, a crippling disease. If we heal the lepers, that's it. We've pretty much done everything of biblical, sort of miracle level at that point. So this can yield all that stuff, and we're getting them. I think there's like 27 approved drugs right now. But that's going to turn from a drip into a stream, into a raging stream, into a deluge. And this is the same feeling that I had in the computer industry, which is you just see how it's proliferating, that it can't do anything but proliferate. It can't do anything but to be like a snowball rolling downhill, because everything is in its favor. And I think that's what we're going to see as well. And I think we're going to see a lot of it right here, because this is where it started, and this is where we have the room to grow and the resources to do it. So, try finding talent in Cambridge now.
Yeah.
It'll cost you 3x what you pay for it down here.
Yeah.
Same thing in San Francisco, right? So I'd like to encourage everybody who's watching this, just move to Maryland, start your company here, because the reality is that you can live a good lifestyle at a reasonable price here, and that drives the cost of everything.
Mm-hmm.
And so, and you can't do any better for talent and access to the FDA and NIH and all these great schools and things like that than you can do here in Maryland, right? So why wouldn't you come to DNA Valley if you want to do stuff with DNA? And I think that, ultimately, hold on to your houses, in that triangle.
Yeah.
Because, we're gonna start seeing housing prices go up. But the good news is we're not bounded by the ocean and and in the case of San Francisco, a bay, where there's hardly any place to grow out. I mean, we've got land as far as the eye can see, undeveloped land. There's still farms, they can be converted into biotech, clusters. So this, could be just amazing, an amazing ride.
Well, I want to share, as we come up a little bit on time here, we do want to allow some of the folks who have joined us here, or if they are, to put some questions in on LinkedIn Live to pepper you with a few, and we'll kind of offer to the folks who are joining us online that if they have a question that if they could send it to us, we'll share it with the AGT team to try and get an answer for them. But I just want to say, like, your vision, what you just said you envision, it's rare to kind of see that embodied in somebody that's actually taking the action to bring it to bear. Your staff knows that we kind of had a dual purpose of wanting to do this fireside chat. Personally, the story that got me involved with AGT, I felt others needed to learn about because I think they could, number one, benefit from it, the awareness of it, the confidence that you're breeding in this ecosystem. We kind of used to talk about exits. I see what you're doing with Adenune as a harvest of sorts, and AGT will continue to plant fields around that seed crop. We have announced at Think, we're doing an event, it's our second annual next year, it's called Think Innovate. We believe if you can stop, think, you can innovate, but you can't stop all the time, you have to keep moving. So iterative success and confidence and results, metrics and milestones. Something that we wanted to do to kind of jumpstart people thinking about Think Innovate is to celebrate the folks who embody that. So we created a Champions of Innovation celebration. It's not an award ceremony, it's not a ranking. It is literally folks who see their peers in this market. It doesn't have to be life science, biotechnology, it could be cybersecurity, it could be manufacturing. We want to celebrate those folks on a year-long run-up to that event and hope they join us there. But what we wanted to do, we just announced it this week, and we felt that what you personally embody, Jeff, in the success of creating AGT and the struggles of building a venture that way, and bringing your first line of business or product to market with the success and confidence you're breeding, we as Think are nominating you as the first champion of innovation for the Think Innovate show for 2024.
Oh, wow, well, it's an honor, and I appreciate it, but I maybe it might be more appropriate to award it to the whole team at AGT and Adenune. It takes a village to raise a drug. I appreciate being recognized for vision and for being the leader of that group, but I think that would be wonderful if you could maybe nominate everybody in the company.
I tell you what,
instead.
You can nominate the company if you so choose, and we will definitely accept that as well as our nomination of you personally. But just another trait that embodies why you're the type of leader that we celebrate in this effort. So thank you and congratulations to you and your team. And for those of you joining us, we hope you will also think about your peers and the communities that you want to celebrate as innovators, champions of innovation. If you go to innovate.com, there is a link to nominate and also understand what we're looking for out of that. We hope you keep an eye on this. Is the first of many connections we want to make with the leaders in this market and the ecosystems that are helping steward and the folks who are covering it. So Chris, I'm going to bounce it back to you that we might have a few questions in the room here. We probably have about 5 minutes, so maybe 2 or 3 if there are any. If not, we want to thank you guys for joining us and keep an eye on this space.
Great, wonderful conversation. So, I've heard your story several times, and every time you tell it, there's something else I take out of it. So thank you. So do we have any questions from anybody here in the audience today? Why don't I lead everybody off with one,
There
just
you
to
go.
get everybody just nurtured a little bit. So, American Gene Technologies, where creativity cures. I feel like innovation was like just seeded in your company from the beginning, but I always love that philosophy. And as a CEO running a company, could you talk a little bit about how do you get that philosophy into the culture of your company so that it's embodied by all of your staff that just got nominated for this innovation celebration? So how does as a CEO, as a leader of the company, how do you instill that into the culture and into each of the PhDs and everybody in the company who are really driving this mission?
Well, I think it starts with the folks you hire. You know, like we always think that an AGT person is somebody that is smart, self-disciplined, in other words, good work ethic, and then has passion, right? So immediately, if you have that energy behind you, then you're going to, you know, maybe not just be thinking about it from 9 to 5, maybe you're gonna be in the bathtub and come up with an idea. And by the way, I was taking a shower in the bathtub. I don't remember the last time I had a real bath. But anyway, I think, you know, so that's sort of the foundation of it. And then I think, yeah, the corporate culture has to encourage that. So hopefully, you know, people in the company see me as approachable and see each other as approachable, and you need to, you know, sort of promote the type of environment where you are happy to have discussions. So at a place like Facebook, they say move fast and break things, right? And I think that, you know, we're not afraid to, you know, come up with crazy ideas that we later abandon, right? And, you know, that's unusual for companies. And so, you know, the more that people see that, they'll do it. And you also have to articulate that to folks. You need to say that, you know, this is what we're looking for. What are we looking for? You know, in those great employees, we want every part of the buffalo, right? You know, we don't want to waste anything. If they have ideas, we want them to get those ideas out. And, you know, it doesn't mean that we can do every idea. It doesn't mean that we'll agree on everything. But, you know, there's a lot more of that out of the box thinking, and we're comfortable with that, and we're comfortable with things that don't work out. So now a lot of stuff does work out because it's just, you know, it's all white space. When we started in gene and cell therapy, like nothing was being done. And, you know, now it's getting a little bit more crowded, but there's so much open territory out there. There's so many great things to do, and it's just a matter of, you know, too many great ideas and not enough time and not enough money for them. And, you know, so that's what limits what we can do. If you look at the history of the company, I was like, well, we got 20 projects going, you know, and okay, what are the ones that would have the biggest impact and the lowest risk? And that gives you sort of a number, and then you arrange it from highest to lowest. That's your Pareto analysis, and then you just go, well, how much money do I have? Well, I can do 3, and that's how we ended up in immuno-oncology, PKU, and HIV.
But with that, I mean, there has to be a little bit of a mixed bag with your human capital that you brought in and invested in AGT, kind of being spun out, a little bit of a team that's going to take that forward, always be connected. But I think there's an opportunity for them to spread their own wings, which any entrepreneur wants to breed the next generation of entrepreneurs from their effort. So I just see that as there's now AGT and a company that you also inspired being spun out that can attract that talent to this region like you talked about, and they're going to have great success.
Yeah, so the actual mechanics of this is pretty much the whole company's going over to Adamune.
Okay.
Okay, because I think that we need to make a success out of Adamune. I'm going to be the CEO of it.
Okay.
And so, yeah, I'll be narrowing my focus a little bit. But, you know, AGT will still be there. I think it's in the lead in terms of creating, you know, a tremendous amount of reusable technologies. We have you know, the patent brag wall that we were talking about earlier. I think that we've out-patented just about everybody in gene and cell therapy, and that has, you know, some value. So we'll get back to that in the future. So, you know, everybody will be narrowing their focus.
Yeah.
But I think, though, you know, we're in a big growth area. So this provides the same sort of challenges that, you know, people will see opportunities in that environment and move up to do new things and grow more personal skills. This is another thing that I always used to, you know, or I always love to say to people that come into AGT is that, you know, working here will make you a more valuable person. So it's not just about the money. I think we pay competitive wages, but you're not gonna get rich off the salary. You know, if you really believe in this in the long term, everybody has stock options, so we're all gonna, you know, get a piece of what we created, so that's really good. But regardless of what happens to you here, whether you succeed or fail, you will be challenged because it's a startup company where, you know, we just throw you in the deep end and go, okay, you know, here's your, you know, 80% job, figure out what you're gonna do with the other 20%, you know, surprise us, right? Swim, and you know, we get a lot of swimmers, and then, you know, those people love that challenge, but the point is that when you come out of that experience, you are a more trained person because we gave you that level of responsibility. You were around other smart, motivated, successful people. That's like it was back in the early Apple days, and we used to say that around Silicon Valley as well. Succeed or fail, you still benefit from the experience of being in these companies that are swinging for the fences. It grows you as a person. And nobody, you know, when a company fails, they all just go over to other companies. That's one of the things that'll be beautiful about DNA Valley, right? Is that, you know, yeah, some of those companies may actually not make it. But it doesn't matter because the people will be valuable in all the companies that are around there. So, you know, I think that the ultimate, you know, sort of thing that I'd like to create at Adimune, and it's not like Adimune can only cure HIV. It can also do a vaccine for HIV. It can also do a better treatment. So there's a lot of different things that could be in there. And then the other thing is what if Excel had come out and then they stopped innovating Excel? Do you think you'd still be using Excel right now, or would somebody else have beaten them? Of course, somebody else would have beaten them. So it doesn't matter if we're the #1 company and the only cure for HIV. We still have to figure out how to obsolete ourselves. Otherwise, somebody else will be glad to do it for us, right? So it'll never end. The creativity will never end. The challenges will never end. If, you know, the HIV cure market is probably a $4 trillion market. That means that even that market is going to go on for decades. Even if we're curing it, it's going to take decades to get to everybody. So, you know, there will always be a challenge. There'll always be an opportunity to take yourself out, you know, on the highway and, you know, and go full speed. And there's going to be a lot of people that love that and a lot of people that gain from that experience.
Excellent. Well, Chris, if you have one or someone in the room has one, we'd welcome one more question.
One more question.
All right.
Bill?
This is another leadership question, and I like what you said about, you know, your vision, but you also said I had to drive this thing, and at the same time you were running out of your retirement. Was there a moment that you remember that you said, oh my gosh, I took the wrong turn? And secondly, how have you evolved as a leader over time?
Yeah, that's a really good question.
Let me just repeat that for the people that are watching. So the question was,
One is, how has he evolved over time as
how
a
have you
leader?
evolved as a leader over time?
And was there a point in time where you were pulling your hair out and said, oh my gosh, I did the wrong thing?
Yeah, I think the answer will probably repeat the question. So, you know, so entrepreneurialism. So the question was, you know, was there a point where you were like, oh my God, did I make a mistake getting into this, right? Okay, so that would be, you know, pretty much every day for the first 10 years. But, you know, sometimes you just keep going because you believe in it. Sometimes you just keep going because you're, you know, if you fail, it'll be a disaster. And so, you know, your demons are chasing you. Right? Whatever motivates you to move forward is part of that journey, because the reality is that the entrepreneurial characteristic is a congenital defect that comes with over-optimism, right? Because nobody who wasn't over-optimistic, who couldn't, like, you know, believe that somehow it'll work out, would ever be able to take those extraordinary risks that come along with entrepreneurialism. So, you know, we might just be gamblers at heart that think that we can tip the game in our favor because, you know, we can use our brains to predict things and whatever. But, you know, it's fraught with implosions and failures, and there were many, many times, you know, over this whole 15 years where I was like, we may or may not make it. Now, that right, means that you have this series of crises, okay? I mean, it never ever stops. You know, at least 3 or 4 a year. And guess what happens? You survive them, and it gives you confidence for the next one. And now I'm like, you know, I can stare, you know, things in danger in the face, and, you know, I'm not feeling it anymore. I may recognize it intellectually that I've got something I've got to figure out here, and it's a big deal, and it may even keep me up all night, but I'm not expending any cycles on worrying, which is really interesting because, you know, you find other people that haven't been in it as long as me or don't spend 24/7 thinking about it. And I mean, even like my wife, right? She's more likely to worry about these things than I am, or, you know, to be up at night worrying as opposed to thinking strategically. I'm trying to just think strategically. But that's because, you know, she wasn't living every minute of it, and so she doesn't even have the data that she might need. You know, she might ask me, how was your day at work? But, you know, the only thing harder than you know, working 12 hours and coming home, you know, having actually accomplished a lot is to then spend the next 4 hours telling somebody else about what you did, right? So, you know, you have that friendly conversation, but the reality is that nobody knows what's going on with the entrepreneur. You know, there's a book out there called The Hard Things About the Hard Things. Whatever. I recommend that to all entrepreneurs. At least you realize that there's somebody that can empathize with you, which is all the other startup people out there. But that actually describes, you know, my life over the last 15 years, and it's pretty good. You know, I think it's amazing parallels and similar lessons. So you know, the how have I grown? I am able to face my fears, you know, strategically. I've had to solve a lot of problems that have made me a more effective leader because you learn lessons about what you're doing wrong, and you need to fix them. You know, including I need to look at me and say, how can I be a better person? You know, that could be anything from communicating the mission better or more often or to the right people. And I used to be really great at selling the company to investors, and then I come back here and I realize the employees don't believe in the company. That's a problem. Right? That they're creating inefficiencies because they're back here just worrying like, where's Jeff? Well, where's Jeff? He's raising the money to pay your payroll. Now get back to work. But they were like, you know, what if all of a sudden you can't pay payroll? I'm like, what if a meteor hits us? Like, if we sit, you know, if we sit around and do nothing, I guarantee you payroll will run out eventually, because we are in a virtuous cycle where we do stuff and I take it out to the market and I show them what we did and I get us more money. It's really tough. So even employees have to grow in that environment, or the leader has to learn who can take it and who can't take it, right? And to make sure those right people are around. So have I grown? I would say, yeah. I mean, you know, this is, you know, I got my street PhD in molecular biology, genetics, you know, virology. I mean, that's pretty good. I play a doctor on TV sometimes. I have everybody calling me up for medical advice. I do not give medical advice. I'm not a physician. Go to your physician. But yeah, I mean, growth, it's like being in school and getting paid for it. And that's what it's like to be at the company in any position. It's like getting your, you know, an education and finally you're doing all the work and you're getting paid for it, instead of doing all the work and then you got to pay the university $60,000 a year or $100,000 a year, right? So, you know, this is a great growth, you know, just venue and opportunity for growth from the CEO to the janitor.
We've seen with whether it's young entrepreneurs or folks who've been doing it multiple times that knowing what you know, but knowing what you don't know and surrounding yourself with people who do is kind of a trait that's bubbled up to the top, and you certainly embody that as well.
Well, yeah, you got to listen to everybody, and so you got to be smart, but not too smart, right? Because I do find a lot of people in business who, you know, have achieved a lot within their area, and so they don't realize that there are a lot of things that they don't know about, even though they have mastered the domain that they spend all their time in. And it's really natural to just stop listening to anything outside of the scope of that. And I face that problem when I'm talking to, you know, experts within my own company, that they're like, you're just a CEO, what do you know? Well, I've been in gene in cell therapy now for 15 years. I've learned a few things. But I've got to make sure I don't do the opposite too, which is like, what do they know about running a company? You know, but they've got questions, they've got ideas, and okay, so you listen. And then also, you know, what you really hope is you don't just listen to experts that tell you what to do. No, you get people in who will grab an area and just make it their own and do something amazing. And I actually want to train my leaders to do that with each one of their lieutenants, who will then create their own lieutenants. And this is a great way to grow a company, because really, what is my office? You know, what is my job is I divide and conquer. I haven't found anybody yet who can, you know, sort of see the vision as clearly as I can, you know, that can articulate the vision and the mission as well as I can, that can go out there and sell this to the community as well as I can. Okay, so that's my job, security. But I can't go over to the cell culture of it and do anything, all right? And so I need great people in that area. And, you know, if you look at, well, what do I need to accomplish as a CEO? There's like, you know, 50 different things. And if I have 50 great people, I can do all that stuff in parallel. And if I can find people that I trust and that trust me in return, and then I can just, you know, basically support them and encourage them and give them authority within the area, and then hold them responsible for the results. So they're like, you know, entrepreneurs within this entrepreneurial company, with the biggest headache of entrepreneurialism removed: payroll. Right? So they get to run a department and have a little company inside this company, you know, if we do it right. And, you know, and we kind of, we provide HR, we provide payroll, we provide benefits, we provide office space, you know, all those things that the universities will charge like a 60 to 100%, you know, overhead on for professors that are getting grants that are at that university. Everybody here just gets it for free, right? So, yeah, I think you know, that's part of the philosophy, and maybe part of, you know, what I've learned, and hopefully some of the things that other people have learned within this company that will continue to thrive and allow us to grow more and more.
Those are the places you'll go.
Yeah, we'll see. We'll see.
Well, Jeff, thank you again, and to your team at AGT, true champions of innovation and entrepreneurship as well. And I also want to thank Chris Thru, and the team at BioBuzz is tirelessly keeping their eye on this industry, inspiring this industry. Kelly Schultz and the Maryland Tech Council just really spreading their wings and bringing that ambassadorship and stewardship of the ecosystem across the state and even across the river and nationally. You talked about a lot of reasons why this is the place to do what you do, and you know, I've put my Maryland Pride hat on as well for a lot of years, and I will continue to do so. But it's not that we want to, you know, talk down other markets. We just have great things going on here. We have great talent and we have great opportunities. So thank you for being an embodiment of that, Jeff.
Yeah, thank you.
And thank you all for joining us. Keep an eye on for other stops on this road show of Champions of Innovation.